Welcome you all to this Cryo-EM course. Last class we already discussed about different types of magnification, how our grid looked like. Now at a different magnification we started from 115 magnification to very high magnification, that is 30K, 36K magnification, 45K magnification, 54K magnification. Based on our sample we can change the final magnification. If our protein sample is or any biological macromolecule sample, if it is a very small, we can image those samples at a very high magnification. If the sizes are very large, so something like bacteria, viruses, generally we go to the smaller size. So generally 4000, 6000, 10,000 magnification. Generally for the tomography, cryo-electron tomography, we use low magnification where because most of the time we are collecting data at data for large molecules like biological macromolecules like bacteria, viruses, cell. But for the protein sample or DNA protein or lipid protein, we have to go high magnification. So here this slide I already discussed in the last class. I want to start from here again because this is showing us that different types of amorphous ice, right? I already discussed. So when we have this kind of ice pattern, if you have a broken ice, broken square like this, so better to avoid this kind of squares, because if z height is not accurate or eccentric height is not accurate, it will be very difficult to get the proper focused image. I hope you remembered all of this. If the eccentric height changes, your focus is going to affect that. You have to pay attention. And then definitely there are some squares are there. If you look at those squares where hole is completely not visible or with a lot of contamination, and this kind of pattern, if you try to image it, most of the time you will end up with a very bad images, or sometime you can see crystalline ice, your particle or proteins or biological macromolecules will not look very nice. So this type of ice, it is better not to use it for your cryo-EM data collection. Now I will show you some crystalline ice, that whenever we are talking about the hexagonal crystal or cubic ice. So here there is a two image I am showing. So here you can see one hexagonal crystal and some kind of bad ice pattern. It may happen because of due to the temperature change or the handling issue. This kind of ice pattern you can see. There is another ice pattern we collected in our microscope where you can see is a very nice hexagonal structure. And also if you look at this ice inside this hole, you can see some kind of pattern here. So it is not very smooth anymore. So some kind of patterns are there even on the carbon also. If you pay attention here, you can see that pattern very well. You can see this pattern very well. You can see this pattern very well in this region. So that is basically a crystalline ice, and this crystalline ice most of the time will give you a very bad image. So if you have a square or holes like that, better not to collect the data in this region. Why this kind of ice we are getting? There is couple of reasons. Because whenever we are handling our grid, we are plunging it into liquid ethane. Then from the liquid ethane we transfer it to liquid nitrogen, then we store it in liquid nitrogen dewar for some time, and then we are taking the same grid to clip it and then insert it in inside the microscope. So there are many steps where from liquid ethane to liquid nitrogen, then liquid nitrogen to liquid nitrogen dewar. From the liquid nitrogen dewar we are taking it out and we are clipping that grid. We are putting it inside the cassette, and then we are putting it inside the nano cap, and finally it is going inside the microscope. So if there are handling issue, if we are not transferring our grid properly from one position to the other position, and if we are very slow, so then because of this temperature change, it is going to change the pattern of the ice. One more possibility, that when the temperature of the ethane is not minus 180, it is maybe somewhere 80 degree, minus 80 degree or minus 90 degree, and we started freezing. So it cannot absorb the heat properly, and then it can form that crystalline ice, uneven ice, what we can see it in our slide. So there are lot of situation where we can get this kind of ice pattern. So whenever we will do the cryo-EM, we have to be very careful to prepare our sample or prepare our grids. So that is one of the bad images I was showing you. So let us see couple of more different kind of bad images. This is taken from a paper where they demonstrated very well that the crystalline ice, how the crystalline ice should look like. So here you can see there is a pattern where in this particular pattern, here you can see there is a some uneven pattern, you can see some uneven pattern also here, some uneven or black patches are there. Here you can see there are some area there is an uneven pattern, and some black, very thick black ice is deposited. So these are the different kind of crystalline ice which is deposited in different position of the grid. So if you have or inside the hole, so if you have this kind of ice pattern, it is better to avoid this kind of grid for further imaging. So previous slide and this slide both are taken from this. So this is the link, if anyone is interested, you can go through this paper. This is a very nice paper that it describes in details that how and what kind of ice pattern you can get it. So here there is a couple of more ice pattern it is showing here. So one of this ice, you can see there is a black patches and then some protein molecules, you can see it here. And now in this particular square, what you can see in this particular square, you can see that here there are some black contamination kind of thing. This is basically individual protein molecules that we are interested to select those protein molecules. But what about those large big contamination? Those are basically ice contamination, or maybe sometime if there is a impure liquid nitrogen or impure liquid ethane, those contamination are coming from those liquids also. Now there is another ice pattern you can see, that is again a very uneven ice pattern where there are some black patches at different position, and there are this is not an uniform ice. So in between there is a microcrystal which is deposited here. So this is another problem you can see because of the slow change of the temperature ice. You can see a leopard skin pattern. So this circular leopard skin pattern, you can see it in your grid when you will image those sample. So from those kind of sample, it is very difficult to isolate your protein molecules. So if your ice, if your background is not very smooth and you are getting different types of ice pattern, and you can see the crystalline ice, better to avoid those kind of images for data processing. So far we have discussed about that different types of images and some kind of bad hole from even at low magnification. You can consider that if the squares are broken or if there is a crack in between the squares, you can plan to discard those images completely. Then I showed you some high magnification images where you can see different types of ice pattern. Now whenever we are collecting data, as we discussed these things previously multiple times, that okay, when we will image, we are generally nowadays imaging as a movie file, not any integrated image. So, if I just go back to here I want to show you couple of, here you can see that okay. So, how we that entire image process image collection thing is going. So, this is our grid, this grid is inside the microscope. Then the first step, if I just consider, this will be the one square. Then in that one square you can see this kind of dots all over. So, because this is a very low magnification, this is 150x magnification. Then we are increasing the magnification and then we can see this small hole, right. So, this magnification we can tell that this is 380x magnification. Then if I go, this is the square where we have entire hole. In this entire hole we are focusing somewhere here, and then we are coming back and collecting the data here. When we are collecting the data, so this one you can consider that this magnification. Then data collection based on our protein sample, this is our data, we are collecting those data, and that may be that 36K or 54K or 72K, or sometime you can go even 100K also. So, based on our protein size we can change. Whenever we are collecting these images, those are basically all images will be movie file. Movie files means those are, I already discussed about this one, that will be that movie. So, that has some individual frames. Now, why those movie collections are important? This is very important because if you look at here, what we are trying to do, if this is our hole, we are trying to collect data here, and this is the area, what is my focus area. Now, first we focused, will come here, will focus here, and this is the exposure area where we are going to collect the data. Now, when we are collecting the data, generally as I mentioned earlier, we are using 40 to 80 electron dose per angstrom square, right. Now, this is a dose, and this may be distributed in 2 seconds, or you can use it for 8 seconds, 10 seconds. So, you are using a long exposure also. So, long exposure, and also you have a strong electron dose. So, you are using a strong electron dose and long exposure time. So, what will happen here, because here you have an amorphous ice, and if you are keeping your electron beam for longer time, it will damage the ice, it will damage the sample also, and there will be a thermal drift, or because of this beam exposure there will be a drift. And if we just collect an integrated image, you are going to restore that drift within your sample, within your image, basically not the sample, within your image all this drift will be stored, and that is going to affect your high resolution data analysis. That is one of the reason we are collecting a movie frame for 8 seconds. So, you just consider that every frame your exposure is 0.2, and then you are collecting some 40 frames, and then, or you are collecting some 20 frames. Then what is happening, every 0.2 second exposure you have one image, and that is how you are collecting another 20 or 30 or 40 frames, and then you are just recalculating what is the thermal drift or drift between this first frame to the second frame, or second frame with the third frame, or third with the fourth with the fifth, and then you are correcting their motion in between their motion, and then you are trying to align those frame in such a way so that movement you cannot notice anymore, and then you are finally giving us an integrated image that we are going to use it for our further study. So, now if you look at this particular image where we have an image here. So, this is our first frame. If you look at this, so this is our first frame where there is some black patterns, we can see it some places, and very small dots, and now we are collecting multiple frames. As I mentioned that we collected almost 20 frames or 40 frames, and then we tried to correct the drift between individual frames. When we correct the drift between individual frames, finally when we corrected them, then we integrated them together, and then we are showing an integrated image. So, here you can see the first motion corrected integrated image here, where you can see this is the integrated image where individual protein molecules are clearly visible here. Those are the black molecules in a comparatively in white background. So, now if we just recall our previous classes, that where we discussed that negative staining images, basically you have a black background and your protein molecules will be white. So, if you see that kind of images, you can consider that this is a negative stain image, because it will always appear in a black background white molecule. Why? Because you are using uranyl acetate or uranyl format, and the scattering will be much higher because of that uranyl acetate and uranyl format, and protein basically carbon, hydrogen, nitrogen, oxygen, and electron beam will pass through the protein easily. So, that is one of the reason your protein will be white, where when there is a very strong scattering, most of the electron will go away because of this heavy metal uranyl acetate or uranyl format, and the background will be black for negative stain. But here it is completely opposite, because in this particular case in cryo-EM we are not using any staining solution. So, we have only buffers. So, buffer is either phosphate buffer or tris buffer or Hipps buffer. Within that we have the protein molecules. Now, if you just compare your buffer and the protein, the composition is very much close to each other. So, they are not very heavy atom, so their scattering will be not that strong. That is one of the reason in cryo images are most of the time are very noisy images, and it will be very difficult to distinguish your sample from the background, because there is no staining solution we are using. Also, your protein, if you compare to your water molecule or buffer, your protein has carbon, hydrogen, nitrogen, oxygen. So, and it is well packed. So, your protein or biological macromolecules is density is much higher than your buffer solution, and that is the reason scattering will be stronger because of that protein, and that is one of the reason in cryo-EM your protein appears as black in a grey background. One more thing, as I mentioned, we are not using any staining solution, so contrast difference will be not there. So, because of that heavy metal staining solution in negative stain, there is a very nice contrast difference between black and white. So, you can easily distinguish your protein molecules from the background, but in this cryo that situation is not there, and whatever buffer solution and our protein sample, they are almost giving the same kind of background. So, it will be very difficult to distinguish the background from the protein molecules. That is one of the reason we are using little bit higher defocus, because higher defocus is helping us to boost the contrast. How it is helping us to increase the contrast, that we will discuss in the next class. But remember that we are using little bit higher defocus. Whenever we will increase the higher defocus, outside of that molecule you can see that white fringe. So every protein molecule, if you look at, so there is a black protein molecule and outside there is a white fringe. So that is basically helping us to distinguish the protein sample from the background. That is giving us that contrast difference, so our eye can see it clearly. So that is one of the reason for cryo-EM we are always using that different defocus value to take images, but there are many other reasons are also there that we are going to discuss in the later stage when we will talk about the CTF correction and high defocus, low defocus, and how it is going to affect our images. Now the next step, it is that when we collected the data we get some kind of 2D images, then definitely next step it will be to look at our power spectrum. So here I am showing you an image. So this is the beam induced motion correction of biological sample. So here you can see this is an image where there are the protein molecules. This sample is not very good because now you know what is called ice contamination. Here you can see some kind of ice contamination also, but those are our ribosome molecules or biological macromolecules. And here you can see this is the frame alignment. There is a motion between two frames and we are correcting the frame, and this is the x and y axis, how much motion we have in the micrometer range that we are calculating, and we are trying to correct that. So this is, if there is a continuous motion, that is going to affect your diffraction pattern as well as your power spectrum also. This is the power spectrum you are looking at, where you can see one of the direction the signal is completely missing. So because there is a continuous drift, and if there is a continuous drift, if you look at the image at your Fourier space, you will lose some of that signal in one particular direction. So if you have a power spectrum like that, so definitely there is either there is a alignment problem, or there is a thermal drift, or there is a mechanical motion or something which is affecting your signal. Now this is the power spectrum you are looking at after motion correction. The same image before motion correction and the same image after motion correction, you can see it restored all the signal, and you can see the signal is almost this range, I can see it. So from the center you can measure this distance and you can tell how much signal do you have, how much angstrom do you have. So if you see more signal at higher frequency, that means you have a chance to achieve a high resolution. So we already discussed about the diffraction pattern and how we are getting the diffraction from the microscope also. And if you recall that, you can see those are the low frequency signal. But if there is a high frequency signal, because if the waves oscillates much frequently, that signal it will go far away from the center. So high frequency signal always stay far away from that center area. And now if you count all this signal, you can see the signal until here. So that means you can achieve this kind of resolution, you have to measure that distance. So from this one I can tell roughly that it may be a 3.5 to 3 angstrom resolution. Now we will go to the next image. So there is another image, you can see it that FFT or power spectrum is completely distorted. In one particular angle you can see there is a problem, so signals are missing, and this is the again motion between that two images, that okay, how the frames are moving at different direction. You can see this kind of motion, and there is no particular way, there is no particular pattern it will follow all the time when there is a motion. So it may be a different pattern. Here you can see this is a very uniform pattern they are following. Here this is a very ununiform pattern this movement is, but after correcting you can see most of the particles very clearly, and this is the power spectrum, and most of the signal is restored after our motion correction. Now we will discuss about the next important part, that okay, so when we are trying to image our sample, there are couple of different possibilities are there. What those possibilities are? So now we are going to discuss about the good image, bad image, or different types of cryo-EM images, that we are going to discuss now. So here I am showing you that first image. So as I mentioned that if there is a hole, we are looking at this hole from the side, what you can see, this will be the carbon layer. This will be the carbon layer. Then again after a distance, so this will be the again carbon layer, then there will be the next hole, it will stay somewhere here. So whenever ice or water, it will stay because of the surface tension, ice water will stay inside that. Then we have to blot and we have to freeze that. Previously I discussed these things, that okay, there are couple of possibilities of ice formation. So there is a one possibility, it is not blotted properly. This is the carbon area. This is the carbon area. When water is not blotted properly, you can see there is a bulge, or there is another possibility that almost something like that. So if you measure this thickness, this is a very thick ice, and now if you consider that you have a very small protein molecules, then there will be multiple layers of protein will stored here. First of all, because of your thick ice, electron beam cannot pass through properly, it will scatter strongly because your sample thickness is very high, and then multiple layers of proteins will be there, and then scattering will be more because of that. So if there is a thick ice, electron beam cannot pass through properly, scattered strongly, and then your contrast will be very poor, and then in this particular case there will be overlap of different projections of that 3D. So if you look at this one, this 3D orientation and this 3D orientation and this 3D orientation is not same. So now if you look at this orientation, you will get a circle. So from this one you will get something rectangular shape something, and then there will be attached. Whenever you look at a 2D projection, you will get this kind of projection. If you look at this projection, what you will get? You will get rectangular shape, two molecules are connected, again there is a rectangular shape, two molecules are connected, then one circle, and then you will get a bulge like that. So when there is a too much concentration and if they overlap with each other and if ice thickness is very high, you will get this kind of bad pattern images where you cannot see your sample properly, and this is an ice contamination, and this may be your sample or this may be our sample, we do not know yet because this is a very thick ice, it is not clear from our images. Now come to the next one. The another possibility is that because of your blotting, if this is your carbon layer, this is the carbon layer, this is our carbon layer, and then we blotted in such a way that this is very thin, and if any protein will stay in this area, this will be in the air water interface, then there is a possibility that protein molecule may break down here. So air water interface, that air water interface is very problematic when you will image biological science sample. So if the ice is very thin and if it is proteins are there, there will be a broken sample. So here you can see there are some kind of dots everywhere, so those are probably the broken sample. And here you can see this particular area ice is very very thin, so we are looking at this particular area. Now if you just go this direction or this direction, ice is comparatively thicker, so most of the protein sample is going to this direction, and that is the reason central part you can't see any particles, only that broken particles because of this air water interface, and then most of the protein moves toward the intermediate region, and that all this intermediate region protein you can see it in this zone. So center you can see a empty area, and then protein is just distributed within this range. And this is the again thick ice area, where this is the thick ice region. So this basically thick ice, this is again thick ice, and this thick ice is contributing here, this thick ice contributing here, or this particular zone. Okay. So next, sometime that larger molecule stay near the carbon area. So here you can see there is a very nice virus particles here, and this is our carbon area, this is our carbon area. So this virus particle is trying to stay near that carbon area. Why it is trying to stay near the carbon area? Because the carbon area we have a thicker ice. If you just measure the ice thickness here, the most of the thicker ice is here, and those large particle will be happy if they will stay near this thick area. And that is one of the reason that liposome or virus particle stay near the carbon area. And here also you can see this is a carbon area, and this is the ice near that carbon area where virus particles are staying. The same thing is here, same thing is here also. So next, another one, that is the moderate ice all over the grid. So you can see the center, it is not, this is the center area, it is not so thin, not so thick. Even the edge area also, it is not that very thick ice. So there you can see a well distributed protein all over the region. Wherever you will go, you can see this well distributed. So this kind of ice patterns are always good, that wherever you will image, you will get a monodisperse sample and the uniform distribution. So if you do a automatic data collection, so you have a hole and you have to just position one particular area to image, and if it is well distributed, it will be much easier for us to image in that kind of grid. So we are always preferred that moderate ice all over the grid, and then we can target different areas to image it. So I'll stop it here today. In the next class we will discuss more about the good cryo-EM images and bad cryo-EM images, and certain other conditions also where we can get a very nice distribution of the cryo-EM image. And then we will discuss about little bit more about the graphene oxide grid and graphene grid, and then we will start discussing about the image processing. Thank you all for your attention.